Welcome to today's episode of Schwegman, Lundberg, and Wolfsner's Chemistry and Biology Seminar Series. My name is Ryan Connell. I'm a principal in SLW's Chemical Practice Group, and I'm your moderator for today. Last year, this series was called the Life Sciences Seminar Series, but we've broadened it this year to include chemistry and to be called Chemistry and Biology Seminar Series, so as to be more inclusive of the chemical sciences that don't always relate to life sciences, such as chemical engineering, cosmetics, material sciences, nanotechnology, and the like. During this series, attorneys from the SLW firm will having a background in chemistry or biology will present on various topics at the intersection of chemical and biological sciences and patent law. We aim to give these presentations on the second Tuesday of each month during the seminar season. If you attend 3 or more of these presentations live and live in the United States, we'll send you a free gift. Slides for today's presentation will be available in the chat, and a link will be emailed to you. As a reminder, nothing in today's presentation should be regarded as legal advice. If you are seeking legal advice, please contact your legal counsel. I'm pleased to introduce our speakers for today, Steve Reid and David Van Vliet. Steve Reid is a principal at SLW whose practice entails the procurement of patents and related counseling in the chemical arts, particularly in the areas of small molecules, pharmaceuticals, and solid forms of pharmaceuticals. He also conducts and advises clients on intellectual property due diligence investigations for transactions including mergers, acquisitions, and licensing matters. A synthetic organometallic chemist, he earned his Ph.D. in inorganic chemistry from MIT, held several postdoctoral research appointments, and served as a patent examiner in the small molecule pharmaceuticals art unit at the USPTO prior to law school. David Van Vliet is a registered patent attorney at SLW. He holds a Ph.D. in medicinal chemistry and has over 15 years of experience in pharmaceutical drug development prior to attending law school. He has experience in most aspects of chemistry, including small and large molecule pharmaceuticals, polymers, and material sciences. His practice involves nearly all aspects of patent application preparation, patent prosecution, and portfolio management, along with extensive experience in freedom to operate, invalidity, and non-infringement opinion drafting. His legal experience also includes due diligence for mergers and acquisitions, along with portfolio valuation and analysis. Today's presentation is titled Patenting Pharmaceutical Solid Forms Revisited. If you have any questions, please put these in the Q&A, and the speakers will answer them during the presentation. That's it. Go ahead, Steve and David.
Thank you, Ryan. Good morning, everyone from the Western United States, and I appreciate that others are across various time zones, so thank you for joining us. And we'll jump right into it. A few years ago, I presented on a similar topic, so today is going to be a revisiting of the patenting of polymorphs. And the first section that I'll be covering will be a brief overview, so I'll be skipping along fairly quickly through my section of the presentation before Dave takes over on the second half. Today, we're to talk about what exactly are solid forms of pharmaceuticals, why they're important, why are they even worth patenting. We'll talk a little bit about some patenting mechanics, such as specification and claim drafting, and then finally, Dave will be taking up the baton for the legal considerations and global patenting concerns. First of all, just I'm not assuming even a baseline knowledge of attendees, so to go over some important definitions, polymorphism refers to different arrangement conformations of molecules in a crystal lattice. So an easy analogy would be water that is in the form of an ice cube on the one hand and snowflakes on the other hand. Both are solid forms. Amorphous, by contrast, is a disordered arrangement of molecules. These do not form any regular crystal lattice, so it's easy to think of these as powders generally. And finally, solvates are those crystal forms that contain some embedded solvent like water or non-aqueous solvent. The taxonomy of solid forms is actually quite extensive. For purposes of this presentation, we'll just refer to polymorphs, but those are really, that's more a generalized term. And as I noted before, solid forms fall into crystalline and non-crystalline forms. Non-crystalline forms are also important to patent. These include glasses and amorphous forms. On the crystalline side of the tree are single component solid forms, and these are what I guess most typically would appear in the patenting realm, such as crystals and nanocrystals. And on the multiple components side would be salts and molecular complexes, such as we have co-crystals and then hydrates, solvates, and so forth. All of these are what we'll collectively refer today as polymorphs because it's just one word to say. Why are crystalline polymorphs important? Well, they factor prominently into regulatory schemes, not only in the United States, but abroad, such as in Europe under the EMA. In the US in particular, the FDA requires uh, uh, generics and um, innovators to conduct polymorph screens and set out fairly rigorous guidelines to do those. In Europe, there's a parallel set of regulations that encourage at least the consideration of polymorphism. It's just enough to know that in both major jurisdictions, polymorphism factors quite prominently in the regulatory scheme. For innovators, obviously, it's a way to polymorphs generally is a way to extend market exclusivity by separately patenting polymorphs over the base patent or the NCE patent. On the generic side, uh, the polymorphs can be a form of non-infringing drug substance. In fact, some generics actually establish their own IP positions. It's important for regulatory purposes that the polymorph, if they're part of an ANDA, have a seamless requirement as the branded product. And specifically, these are pharmacokinetic and pharmacodynamic parameters. Now, another way of emphasizing the importance of crystalline polymorphs is to examine their prevalence in Hatch Waxman litigations. And here we see about a 25-year survey of filed Hatch Waxman cases over the years and from 2025, and about 250 to 350 cases have been filed per year in their most recent years. And of those, about a few percent and increasing to a pretty steady 20% of those Hatch-Waxman cases involve polymorph patents, and a substantial number of these cases were those in which the polymorph issue was dispositive, not just an ancillary concern. And legal considerations in these Hatch-Waxman litigations were infringement, obviously, and obviousness, anticipation, inherency, enablement, and written description. We'll come back to these legal doctrinal considerations in the last half of this presentation. So another way of emphasizing the importance of polymorphs is to look at some cases, specific cases. In the case of ritonavir, which is an HIV/AIDS protease inhibitor, it was discovered 2 years after the launch. A new polymorph had formed during minor adjustments in the production of the drug. And what happened was the second polymorph was had much lower solubility than the intended original polymorph, which was a disaster in the medicinal therapeutic context. In the case of rifaximin, which is a broad spectrum antibiotic, two polymorphs were known, and each of these had very different in vivo pharmacokinetic profiles because of their differences in solubility. So if the dissolution of one is too slow, it could fully transit the GI tract and be excreted before it exerts its full therapeutic effect. Consequently, the differences in solubilities between polymorphs can have fairly profound effects in a an innovator's decision to formulate extended release or immediate release formulations. For those, I won't spend much time on this slide, but for those who want to grab our presentation materials afterward, these are some or reference materials to show further details on the case studies of polymorphism. Now, into some mechanics. Again, I'm going to skip through these fairly quickly since I covered these a few years ago, but it's a fairly data-heavy application that's going to be the one that wins the day, not only for obtaining a patent, but potentially in enforcing a polymorph patent. So it's important to describe, for instance, advantages of the polymorph. They will certainly emphasize this later in the legal doctrine section. But these would include the ease in making the polymorph, how it's formulated, is it hygroscopic, what its shelf life is. Why is the polymorph pharmaceutically important? Well, many polymorphs can undergo various equilibrium conditions to other polymorphs. So physical stability is important. Also, the chemical stability, where they degrade. Some polymorphs are relatively stable as to air and water, while others are not. Solubility profiles are incredibly important, as I mentioned before, in the context of immediate or extended release formulations, and assays that would illuminate PK/PD profiles are also important. Biological data, often which is obtained at the stage of polymorph screens because of the stage of drug development, are very useful. In fact, sometimes dispositive of patentability concerns. It is useful, in fact, encouraged to draft the application where the polymorph under question is the one which is destined for the marketplace, considering whether the polymorph will be an infringing polymorph that is contemplated by generics, which underscores the need for fairly thorough polymorph screens up front. It's also useful to consider that even if the final drug product is a liquid or syrup formulation or the like, polymorphs often are considered to be intermediates to that final drug product for purification purposes. And so protecting the polymorph is a way to block competitors from and their synthesis route to a final drug formulation. How are polymorphs characterized? Well, in the case of the composition, is it a single API, salt form, solvate, and so forth? How is it made? These are recrystallization conditions, which have to be fairly explicit in the application. How are the chemical physical characterization includes, for example, single crystal diffraction in some cases, but the gold standard is the time-honored powder X-ray diffraction. Physical properties are also important. These include, for example, melting points, dissolution rates, and also thermal properties, DSC and TGA, which are data that is sometimes inhabits patent claims. Additional techniques would be solid state Fourier transform, infrared, solid state Raman. As I mentioned before, some polymorphs can exist in an equilibrium with other polymorphs or non-equilibrium states. So it's important to characterize which polymorph they transform or convert to another polymorph and under what conditions. Claim drafting is a skill that should be developed with particular expertise here. And one of the ways to view a polymorph patent is as a rifle shot. The claims are actually quite narrow, but there is a point at which they can be too narrow, which I've described here as a needle. The claim can't be a needle. We'll look at an example of that shortly. It's important to realize that polymorph patents aren't the typical NCE patent where the genus claims would be akin to a shotgun blast, broad in scope. Such claims, generally, if they're drafted well, would have durability across jurisdictions, but there can be some country-specific requirements, which we'll get into momentarily. Here's an example of the needle way of drafting a polymorph claim. This is from Glaxo case back in 1996. The information here we took from the district court litigation, although it went up to appeal and it was affirmed. The claim 1 was directed to a form 2 polymorph, where that was characterized by 31 distinct peaks in the IR spectrum. This is the main claim. Claim 2 further added to this by reciting additional, X-ray powder diffraction peaks, which was in the claim itself further required intensities and obtained by a certain method and a camera and radiation type. Very, very narrow. And the district court judge pretty much laid out why this was a claim destined for failure. It was impossible to compare or satisfy the all elements rule of this claim, where the allegedly infringing polymorph was just a single infrared peak. So the lesson here is don't stuff a main claim with so many peaks and data. Claim drafting for polymorphs can be likened to dactyloscopy, which is the study of fingerprints. And in fingerprints, we use to not only identify a person, but also to distinguish one person from another. So too would be the claims directed to polymorphs, which must focus on or establishing the identity of the polymorph, but we don't have a way to describe the entire polymorph, just as we wouldn't refer to a person by every single feature of that person. Practical way of envisioning this dactyloscopy for polymorphs is by reference to an XRPD spectrum here, where I've picked out a few different peaks and likened those to the individual features of a fingerprint, arch, a whorl, and a loop. And so the challenge for the patent attorney is to select just enough peaks that would differentiate between polymorphs and to identify that uniqueness of the polymorph. And this kind of identification by reference to XRPD peaks is what courts should be able to use, patent examiners should be able to use, and in general, the person who is of ordinary skill in the art should be able to use. Now, it's important that sometimes to realize that in the case where an API has many polymorphs, crystalline polymorphs, to distinguish between those. And one way to do that is to stack diffractograms of these different polymorphs, align their 2 theta, that is the x-axis, and then identify the peaks that might be common or almost common to each of the polymorphs. And in this example here, which is just 2 of over 70 polymorphs of atorvastatin, the red lines show that at least with these 2 polymorphs, there's similarity between 3 peaks. And so a main claim reciting just 3 XRPD, these 3 XRPD peaks wouldn't distinguish and identify these 2 polymorphs. However, that would be a claim that is generic to both polymorphs. The blue lines, in contrast, would be sufficient at a main claim to uniquely identify, in this case, polymorph A, the top trace, and distinguish it from polymorph B, the bottom trace. In an ideal claim, it should identify the molecule, that is the chemical itself. This is the NCE, usually the drug candidate at this point. Identify a radiation source and wavelength, the X-rays, state experimental error, and the claim elements itself could be supported by further data in the specification, such as DSC and TGA data. Here are two examples of a typical garden variety polymorph claim where the crystal form is identified as a crystal form with the name of the API, and it has an XRPD pattern reciting, in this case, 3 peaks of a certain wavelength with experimental error showing the radiation source and the wavelength associated with that source. The second example of claim 1 recites signals. Now, there is a potential difference between a peak and a signal, whereas a peak is defined usually by a peak picking algorithm, the software of a diffractometer, and it's computer connected. So that lining up an XRPD pattern with allegedly infringing XRPD pattern must be peak to peak. Signal, on the other hand, it requires only that some signal above, say, a 3-to-1 signal to noise ratio is present, so that a signal might account is a little squishy and broader and could capture an infringing XRPD pattern where experimental conditions and slight differences, maybe contaminants in the sample, would shift the signals just a little bit. Both methods of claiming could be used and should be distinguished in the specification as to what a peak is defined and how a signal is defined. This slide shows typical dependent claims that simply recite further distinguishing and characterizing peaks of each polymorph. It's important to realize, in the case of atorvastatin, that at the outset, a polymorph screen might not have identified all polymorphs, which might underscore the need to use the word signals in a claim instead of just peaks, or at least have both kinds of claims present. And finally, as a further potential layer of claiming to consider would be a product by process claim. In this case, it's a crystal form where it's prepared by and then a process that is the recrystallization conditions used to make the polymorph. Without getting too much detail, these kind of claims could potentially invoke in the US a 271G grounds for infringement, where an infringing process occurring overseas is an infringing act in the US for infringement purposes. And we reach a point of the slideshow where I will turn over to my colleague, David, to discuss legal doctrines.
Thank you, Steve. The legal doctrines are going to be very similar whether we're dealing with your traditional small molecule genus or if we deal with a crystalline polymorph. They're going to be the same ones. They just have a context that is very specific for polymorphs. Anticipation and novelty, obviously, we have to have that. In most instances, though, we're going to find that the molecule in question was disclosed in a broad genus several years earlier. The polymorph screen isn't done at that time. But if that initial disclosure included, say, in vivo data, something that was more advanced in the process, you might have prepared multiple grams and recrystallize it. So we've got the danger to look out for possible inherent anticipation. Now, obviousness, obviously, we've got to deal with that. Right here with obviousness inventive step, we're going to see a difference between jurisdictions and how they play out. And we've got a couple of key cases that illustrate this very well. Inherency, well, not all molecules have a crystalline polymorph, so it may or may not be present. They require special conditions to form. They do not form in nature. And so what happened in the prior art may have inherently disclosed it, but it wasn't characterized. So caution in the earlier application is also warranted. This is one place where I think the illustration of inventive step versus obviousness is most apparent. Obviousness focuses on the final destination. Where are we going? What do we have in the end? Inventive step focuses on the trip to get there. Where do we start and how do we get there? What steps do we take? The ending is usually not the key point in an inventive step analysis, whereas in an obviousness, it is. And so we have to pay close attention to our jurisdictions. If we're in the United States, obviousness is the jurisdiction, or I should say, is the issue. If we're in Europe or many other countries, it's inventive step. So the application has to take this into account.
Yeah, David and Steve, we have a question in the Q&A. It's a request to discuss Orange Book listing of polymorph patents.
Okay. As a quick note, Orange Book patents in Hatch Waxman litigation, as most of us know, they are over Orange Book listed applications. Polymorph patents are Orange Book listed, and they are frequently the one patent that's available to get your 30-month stay at the FDA. And so being able to list them, absolutely, yes, you can, and critically important that you do so. Hopefully that tells us what we need to know on that, but unquestionably, the importance cannot be underestimated. Because if the polymorph is the only thing left protecting you, then that's the one that's going to get the 30-month stay in Hatch-Waxman litigation. Grunenthal, the molecule Nucenta, we've got a very traditional claim. A crystalline form, we've got it very well identified by name. We've got the X-ray lines with the 2 theta. They've got the radiation type, and they included a few more peaks than I would have put in the original independent claim, but it's a very well formatted claim. The challenger argued obviousness. Now, the references they use, especially the Byrne article at the last bullet point, this is where we very much diverge in inventive step. The challenger argued, well, the FDA says you have to do this. It is an automated process. You can find a contract lab to do polymorph screens. It takes a few weeks. Why is that not obvious? And this is where we now diverge. The Federal Circuit came back and said, it's a routine method. Can we take a routine method to produce something that's non-obvious? The Federal Circuit said, or I should say the district court said yes, and the Federal Circuit agreed. There's no reasonable expectation of success when you're making or finding the claimed polymorph. It may or may not be present. We don't know until we have it in hand. Now, Grunenthal left open the obvious answer that maybe polymorphs in general could be found obvious. But at the moment, and this was several years later, we can look at how they're still applying the law. And with rifaximin, the district court found that I would say that the brand owner made an early mistake. The prior art disclosed the claimed polymorph, but as the court said, the difference between the prior art and the claims is nothing more than the routine characterization to identify the polymorph. They did not use the phrase inherent anticipation, but that's my interpretation right there. They did not come out and say, it's a polymorph, it must be obvious. They went to a different doctrine and did not use inherent or did not use obviousness. They went through something that was almost inherent anticipation without cleanly saying the magic words. The molecule was formed. They recrystallized it using the same exact conditions, but the prior art didn't characterize it. They didn't do the XRPD or any of the other characterization forms. So the court said it was not novel, but they emphasized how it was not characterized in the prior art rather than the lack of novelty. They got to the right conclusion, I think, but they could, I think, have done a little better job in being clear that this was about inherent anticipation. So an early disclosure in that genus, the purification and isolation can get you in trouble if it's disclosed. If all you have is, say, column chromatography and you get the amorphous form, okay. In the United States, at the moment, we're still okay on this. But as the Grunenthal case said, maybe that changes. They left the door open. What they did is they left the door open to go down this particular pathway. In Europe, case 777 basically killed polymorphs for the sake of polymorphs. This was an appeal of a revocation during an opposition. The patent issued, it was challenged in the opposition period. It went up on appeal, and in the end, the board issued a very telling, important decision. There was agreement at the beginning that the patents on the original API, the PCT, were the closest prior art. There was no argument there. It's also known in the art that amorphous forms are not good for formulation. They frequently have flowability problems. They don't dry as well. They have other issues. It is also well known in the art, again, crystalline forms have different physical properties. Those two particular properties are known to be better in crystalline forms. This is, again, well documented. So it is expected. It is not an inventive step to go looking for that improved property. Now, the patent proprietor tried to say, well, there's going to be bad properties associated with that new crystal form. The board came back and said, of course, there's going to be different properties, but there's going to be a trade-off, but that does not change where you're going to go to look. And then on a second line of reasoning that I think was even more to the point. Again, the same closest prior art. Polymorphism, it's known in the prior art. Your innovators in the US and Europe are both required to investigate polymorphism. Screening is well known. It's virtually automated. That alone says there's no inventive step. And so when the final conclusion came out, in the absence of any technical prejudice, the mere provision of a crystalline form is not inventive. And they took a jab at those of us who write patent applications. We all like to write the phrase, it has been surprisingly found, and we're all guilty of doing this. But contrary to the statement in the patent at suit, it is not surprising. And they took a small jab at all of us, and I think we all agree that we've all been guilty of writing that before. Now, the question, though, is what references were cited. And this is where it's very critical. The three references, Bavin, Byrne, and Hancock, were cited in the Grunenthal case in the US. Same references. Whoever wrote the brief in Grunenthal, I am pretty sure read this case because the arguments mirrored it when you read the briefs. But in the US, Grunenthal said, okay, it's obviousness. In Europe, it's inventive step. It's the destination versus how you take the trip. So these references had nothing to do with the torvastatin. They do not mention it. Bavin, we're talking about process development, large-scale synthesis. Byrne, regulatory considerations. Hancock, we're characterizing and talking about the amorphous state in pharmaceutical systems. Very general references that emphasize this is an established, expected route of development for pharmaceuticals. So absolutely critical is how these references were used. But as they always say, the rubber hits the road. How does that work? Well, shortly thereafter, the examining division rejected the patent on another API. They cited in part to 777 in the rejection. However, the rejection was reversed. And how it was reversed and why is the absolutely critical thing to understand. They emphasized the technical problem and how it was addressed in the claims. The specification was very clear. The technical problem was explicitly defined in the spec. The provision of the API having an improved dissolution rate, having improved bioavailability, hygroscopicity, and good stability. There was supporting data in the examples from multiple polymorphs. They were multiple polymorphs were discovered, they were tested, but only one was claimed. And the conclusion from the board that upheld the claims, or I should say, reversed the examining division so that this patent could issue. The mesylate salt claimed would not have been expected to deliver the desired combination of properties. We also would not expect an arbitrary crystalline salt to be suitable in this respect. It's the combination of properties. It is not a polymorph for the sake of a polymorph. So the key point, I think, in 777, it was a polymorph. That's all. In 643, it was, we have a very specific technical problem. We're trying to address that problem. Here's how we did it. And it comes down to, and this is in other cases also at the board in Europe, the patent owner has to control the technical question. If you let the examining division or the board define the question, they're going to go as simple as possible. They're going to say it's a formation of a polymorph. If they control that question, you're done. The patent will not be issued. The claims will not be upheld. But if you can get into the specification, the clear statement of what the technical question is, if you can get into the briefing ahead of early, the technical question is, here's what it is, here's why it is, here's data that supports it. And data illustrating both successful and unsuccessful attempts to solve the problem are especially strong. Again, it's the combination of properties and a problem to be solved. Once you get outside of Europe, I forgot about this. The last question in Europe, and again, we don't know the answer on this one, unfortunately, nobody does. Is the UPC going to change anything? We don't know that answer. So far, I have not seen anything addressing crystalline polymorphs in the pharmaceutical space, and I think it's too early to tell. Early points from different firms and different decisions over there say that they're going to adopt very much the same ideas that were originally through all the pre-UPC stuff, but stay tuned. Right now, nobody knows how this is going to play out. In Japan, we go back to atorvastatin. This molecule was at the center of a whole bunch of decisions, and it illustrates the worldwide impact on a crystalline polymorph, not just the US, not just Europe. There was litigation, even though we don't talk about it, in other jurisdictions. But Japan, the appeal board at the patent office said it was inventive. We were allowed. The IP High Court reversed. They walked down almost the exact same logical line of reasoning that was used in Europe in 777. The improved properties are highly predictable. Your skilled artisan is going to obtain crystalline forms. You're going to do this. So, no inventive step. The key point is trial and error crystallization is not inventive, if that's all that's going on. But the phrase they used, which is similar in the US to, say, unexpected results, does the claim invention have remarkable effects? And they don't explicitly define what that means, but they're going to be looking at the data. They're going to be looking at how the polymorph is superior in one or more properties to the other known forms. Is it superior to the amorphous form? Is it superior to other crystalline forms? Japan also illustrates another important point. Post-filing data is difficult to get in. I'm not going to use the word impossible, but it's very difficult. Canada is another jurisdiction. Post-filing data is difficult. So are many other jurisdictions. If you need to argue over those remarkable effects or those unexpected results, the data's got to be in the specification. If you don't have the data available at the time when you're thinking about writing it, it's definitely possible that you may not be able to get the data in, or you should delay filing. I saw a question just pop up about post-filing data in Europe and Japan, or Europe and China. I would say that China can be more difficult than Europe, but it's not easy, but it's not impossible. Europe, it's easier, but it's not automatic. And so anytime you have to rely on post-filing data, you're opening yourself up to a landmine. You may not get what you need. And so if you don't have the data, you don't have the data. There's nothing you can do, and you're going to try. And you're going to say that we've hinted at this and we've suggested at it based on hopefully an appropriate description in the specification.
Dave, I
Whether
would
they,
add that
okay.
in China, it's difficult but not impossible to get post-filing data introduced into evidence. Historically, that is true, or rather, it's admissible if the application itself contemplates the category of data that you would later present during prosecution. That's a pretty difficult hurdle to overcome. In Europe, by contrast, it's quite permissible. In fact, data can be just presented on the record by the attorney. No special affidavit is needed, as it is in many countries, including the U.S.
Okay. China, right now we have somewhat of a conundrum going on, and nobody knows the answer, and the law firms and people we've spoken to don't know the answer. In a recent case, the Patent Office denied the application using the standard polymorph screening as routine. The standard at the time was unexpected technical effect with clear superiority over the prior art being required for patentability. But the Beijing IP court reversed. In China, there are 3 IP courts: Beijing, Shanghai, and Guangzhou. Beijing is sort of like I don't know, call it the favorite stepchild out of the 3, but above them is the Supreme People's Court. The Supreme People's Court, akin to the U.S. Supreme Court, sets the precedent, and the lower courts, the 3 lower courts, follow it. But the Beijing IP Court came out and said a better technical effect is sufficient. And examples they used: practical usability. That one's relatively straightforward. Same with commercial viability. But another one they included has me a little bit bemused. A significant investment in finding and developing the polymorph. That one, like I said, has me a little bit bemused. And so this could be considered a very significant doctrinal shift. And here's where it runs into the issue. The Supreme People's Court sets the precedent. When they say it, it goes. But why did the Beijing IP Court not follow it? Where is this change coming from? Could it be an internal discussion? Could it be something else? We don't know the answer. We also don't know how this is going to play out at the patent office. There have not been enough patents that have come up or come to attention on crystalline polymorphs that where this could be, we could start seeing an effect. Is this unexpected technical effect going to matter? we don't know. And so it's just going to be a matter of how it plays out over the next several years. And again, it's something that we've definitely got to pay attention to, because why they did not follow the Supreme People's Court, again, has me very curious. In India, is notoriously unfriendly to innovators. Once you get an allowance, there's a pre-grant opposition period. Your putative generic manufacturers are frequently challenging things. In this case, they challenged Gleevec. The patent office rejected the application. It went through the high court. It went to the board. It eventually ended up in front of the Supreme Court, the Indian Supreme Court, obviously. And there it ran straight into the statute. Section 3D of the Patent Act in India basically says the new form of a known substance without enhancement of efficacy is not an invention. And they specifically call out salts, esters, ethers, and polymorphs. Salts and polymorphs are very easy to understand. That can go under the same rationale as the 777 in Europe. But esters and ethers implicate prodrugs. Prodrugs may or may not be the same. With the prior art patents, we had a general discussion of pharmaceutical salts. It was, here's a molecule or a pharmaceutically acceptable salt thereof. And that was sufficient to disclose the claimed composition. Now, the polymorph at issue was identified as the beta form. It had a very significant increase in bioavailability. And here's where it is sort of confusing. Bioavailability was deemed not an improvement in therapeutic efficacy. But an enhancement in efficacy, if we're going to improve the therapeutic effect, the same molecule is always going to have the same therapeutic effect. So we would have to have a different molecule to get a different therapeutic effect, and a different therapeutic effect with a different molecule where we now have a situation where you're not claiming the same thing anymore. And so the general hostility towards innovators is going to say pretty much that polymorphs are going to be very difficult because they're going to have the same biological effect once you're in a cell culture type situation. Your PK, your DMPK parameters, your Cmax, your AOC, those are not sufficient to support a polymorph in India. Now, they can have a different biological effect because we could have different dissolution rates, but that's not an improved therapeutic effect. So it's a difficult situation, I think. And so I don't see that becoming any friendlier anytime soon. The next couple slides are sort of a summary, and we decided to test AI is the most recent buzzword that a lot of people are using and attorneys are getting themselves in trouble over. I started asking questions about crystalline polymorphs in jurisdictions where we knew the answer. So we worked very slowly to develop a prompt to look at different jurisdictions. And using 3 of the different large language models, we're able to come up with very consistent, agreeable effects that were accurate in the major jurisdictions where we knew the results. And so after that, we had the same large language models do a survey of all of the patent jurisdictions. And we were able to verify in most cases that the information was accurate. So what we're finding in a lot of places, a technical effect is required. In some cases in the UK, there's a flexible approach. If we look at jurisdictions in Southeast Asia, unexpected results, Taiwan, strong data requirements, Philippines, data-driven. What we're finding is in many, many jurisdictions, the inventive step, I would call it paradigm, that you see in Europe is the primary driver. So if we prepare a patent application for the European system, we're going to have good data for wherever we want to go. Does that mean we can get it in other places? Obviously, we're going to run into the variations on those laws. And so what we did is looked at from a very, call it the heat map type of thing. Green, we've got a good chance of being able to get one, red being more difficult. India, well known for being difficult. South Africa has a registration system, so obviously that's as easy as it gets. United States, pretty straightforward. So you've got a different level of difficulty in many cases. But what I find, I would call it comforting. I can focus on writing for Europe and the United States and know that I'm going to be covered for almost all jurisdictions. Um, will it guarantee success? Well, that's impossible to say. So, key practice tips. Um, for some reason, the global plan ended up second on here, but that's where you start. Where are we going to go? Identify your jurisdictions. Novelty is often not enough. In the US, novelty often drives the allowance, but in Europe, Japan, China, no. Novelty is insufficient. We need hard, tangible evidence. And the more data we can include in the application, the more we are positioned for different jurisdictions in the world. Again, now, once we start collecting data, the better quality data, the better. If we've got good, clear, reproducible results, good signal to noise on our XRPD, that's going to enable us to have something that's more enforceable and more readily identified. Because when you've got cloudy, fuzzy data, it's the case of garbage in, garbage out. We've all dealt with bad data. It makes for bad claims. But if we've got good data, good information, we can come up with something that is easily or more easily defendable. Once we're ready to write, now on the next slide, claiming strategy. We need overlapping and narrowing layers of claims. This is how we write a genus for small molecules. It's the same thing. We start broad, we focus down. So with the XRPD, we may claim 3 peaks in the independent claim, and in a dependent claim, we might add 3 more. Another claiming style that's used upon occasionally is we've got 3, the independent claim, we have 3 of the following 5 peaks. There are several different styles, but we're going to use an overlapping, narrowing layer for protection. We're also going to have data for how it's formed and how it's recrystallized. Whether we include that in the claims right away or not, it can add another layer of protection. Product by process claims can be very useful. Also, as always, we need to know the prior art. Did someone inherently disclose it? If they have, we've got to be able to be very careful on how that happens. So even before you've done the prior art or the polymorph application, we probably need to know what's going on in those early genuses. And we've got to be able to distinguish it from the prior art.
David, I would add that it is often the case, but not always, where the drafter of a polymorph patent application is also the drafter of the earlier
Yep.
NCE application, which in most cases is stuffed with many synthesis examples. And so, barring any third-party prior art, the drafter in my hypothetical is the controller of the closest prior art, that is the NCE itself. And so a good tip in going through the example section is to either have the writer of the examples, sometimes it's or a contract research organization, or maybe the inventors themselves, scrub the examples of exact details on isolation, which in no way underscore or undermines enablement of the compound itself. So, for instance, you would not want to say Compound 4 was obtained as colorless cubes by recrystallization from ethyl acetate and toluene at 20 degrees Celsius over a certain number of days to obtain 51% yield. Now that sounds great for a journal article, but that would pretty much sink all chances of patenting that inherently disclosed polymorph in the future.
And Steve, a question just popped into the chat box. What if there's a prophetic disclosure, meaning in silico draft in the spec, and then we submit post-filing data during prosecution? It says the question is more general and not just in relation to the presentation, but how is that viewed in Japan, China, and Europe? With a polymorph, I would say a prophetic example isn't going to do anything because in general, it's prophetic. You don't know anything about what the peaks are. We know in Europe and inventive step jurisdictions, of course, we're going to do the screening. So we're going to predict, okay, yes, it recrystallizes. But in the US jurisdiction, we don't, okay, you've made a prophetic example, but you don't have any prophetic way to anticipate what the XRPD spectra is going to look like. I mean, there's no way to predict that I am aware of computationally. So there's not going to be a predicted spectra. So from the peak picking point of view, a prophetic example isn't going to say anything.
Another way of viewing this
And
is,
so do you want to?
sorry, I didn't mean to cut you off. I was going to point out that in the U, I realized the question wasn't about U.S. specifically, but here, invention is conception and reduction to practice for a given molecule that typically is a target molecule written on a whiteboard and at least some idea of how to make it. And then it's actually reduced to practice by carrying out the synthesis or constructively reduced to practice by filing the patent application. The polymorphs are really different because you don't have an invention until the exact point in time when you have a crystal in your hand. So the inventing process is sudden and unpredictable, as Steve was pointing out. So in the other jurisdictions, I believe a prophetic disclosure is probably no more damaging than the closest prior art, which contains a description of the NCE and then with a raft of other references that describe generally how to make polymorphs. The fact that there may be an in silico data probably strengthens that prior art scenario, but in principle, the challenge for patenting is the same.
Yeah. And from the technology point of view, I actually have not seen any kind of good in silico work that says this molecule will recrystallize in this single solvent or combination of solvents. I haven't seen any in silico software that is valid for that kind of work. And so you'd be throwing darts at a board, pick your two solvents and hope they're miscible and hope you crystallize. So any kind of prophetic example, I think, would be you wouldn't really need to worry about it, I think.
Okay, well, I think that's just about our hour.
Yeah.
Yeah. Well, Steve and David, thank you so much for the excellent presentation. To the audience, if you have any other questions that occur to you after today, please feel free to email myself or the presenters. And just as a reminder, there will be a link to today's presentation that'll be emailed to you. I hope you'll tune in for additional episodes of our SLW Chemistry and Biology seminar series. We're done for the season, but we'll resume in September. So please stay tuned for that. And thank you all very much for your attention today. And thank you again, Steve and Dave.
Thank you.
Thank you.